All right, let's get right into it. Today, we're doing a deep dive on a microbe that, often flies under the radar but is becoming a real issue in clinical settings, Aeromonas-Caviae. It's way more than just some random bug in the water. It's a pathogen that you really need to have on your radar. And this quote from our source report really just nails it. It perfectly captures this bug's journey, right? From being a common organism you'd find in a pond to a major player in some pretty serious human infections. This is the story of how we're being forced to rethink the role of Aeromonas-Caviae. Just so you know, all the information we're covering today comes from a comprehensive report put together by Gemini Deep Search. For all of you who want to really get into the weeds, we've linked the full notebook document. It's got all the citations and raw data right in the description below. So here's the game plan. We'll start with why it's a growing threat. Then we'll jump to the bench to see how to spot it in the lab. After that, we'll cover the diseases it causes, the absolutely crucial details on resistance and treatment, and finally wrap up with how to stop it from spreading in our hospitals. First up, let's figure out where Aeromonas-Caviae actually fits in the microbial world. Because, let's be honest, accurate ID and understanding its behavior all starts with getting the taxonomy straight. So, for the longest time, Aeromonas species were kinda lumped in with the Vibrionaceae family. But now they've got their own family, Aeromonas-associated. And this isn't just shuffling names around for fun. It reflects a huge ecological difference. Aeromonas loves fresh water, while Vibrio is a saltwater bug. This split points to a completely different evolutionary path, which directly impacts its virulence factors and, importantly, its resistance patterns. Okay, let's bring this out of the textbook and onto the lab bench. How do we reliably identify Aeromonas-Caviae and, more importantly, not mistake it for something else? On a gram stain, it's a pretty classic-looking gram-negative bacillus. You could easily mistake it for one of the common enterobacterales. But here's where it gets cool. It has a dual flagellar system. In a liquid broth, it uses a single polar flagellum to zip around, but put it on a solid surface, and it starts expressing lateral flagella all over its body, which lets it swarm across the agar. A pretty neat trick for colonization. Now, on the culture plate is where things can get really tricky. On blood agar, Aeromonas-Caviae is usually non-hemolytic, which is a key difference from the very beta-hemolytic Aeromonas hydrophila. And here's the classic pitfall. On MacConkey agar, it often ferments lactose, turning the colonies pink, making it look for all the world like E. coli. This is exactly why the oxidase test is non-negotiable. And of course, no growth on TCBS agar. That's your quick and easy way to rule out Vibrio. So, how do we nail the ID? It really comes down to a few key biochemical tests. The positive oxidase test separates it from the entire Enterobacterales order. Resistance to the vibriostatic agent O129 separates it from Vibrio. But within the Aeromonas genus itself, the single most useful test is the Voges-Proskauer, or VP. Aeromonas-Caviae is VP negative, while its more notorious cousin, Aeromonas hydrophila, is VP positive. It's a simple, elegant, and powerful differentiator. Okay, so we found it in the lab. What does it actually do to people? Well, it turns out Aeromonas-Caviae has a pretty distinct clinical profile. And this chart just says it all, doesn't it? I mean, look at that massive slice of the pie, pediatric gastroenteritis. In some studies of kids under 5 with diarrhea, Aeromonas-Caviae accounts for something like 60% of all Aeromonas isolates. It's also a big problem in liver and bile duct infections, bacteremia, and, of course, skin and soft tissue infections. And if we dig in a bit, the diarrhea it causes in kids isn't always a simple, short-lived bug. It can become persistent, watery, and even dysenteric. In adults, you'll often find it as the culprit in hepatobiliary infections, especially if there's a blockage from a stone or a tumor. And when it gets into the bloodstream, it's typically hitting patients who are already really sick, like those with cancer or severe liver disease. All right, now we get to what might be the most critical section for day-to-day practice, antimicrobial resistance. And for Aeromonas-Caviae, the whole story really hinges on one very important genetic exception. First off, let's talk intrinsic resistance. Aeromonas-Caviae comes right out of the box with a chromosomal AMP-C beta-lactamase. This isn't something it picks up. It's just part of its standard genetic toolkit. And this means your common go-to beta-lactams, ampicillin, amoxicillin clavulinate, your first and second-gen cephalosporins, forget about them. The bug just inactivates them. But here is the absolute game changer, the single most important therapeutic differentiator you need to know. Unlike Aeromonas hydrophila and Aeromonas veronii, Aeromonas caviae almost never has the intrinsic metallo-beta-lactamase gene, cphA. This gene codes for a carbapenems, an enzyme that chews up our big gun antibiotics. The fact that Aeromonas caviae lacks it means that carbapenems, like meropenem and imipenem, are usually effective. This is a massive, massive distinction. For all the labs out there following European standards, here are the official EU CAST clinical breakpoints. This is the rulebook. Having these standardized numbers is absolutely essential for making sure our lab results are consistent and clinically meaningful, no matter where they're being generated. So, what does this all mean for treatment? Well, if you look at both the Dutch and international guidelines for severe diarrhea, there's a pretty clear consensus. Fluoroquinolones like ciprofloxacin or cotrimoxazole are the recommended oral options. For kids, you're generally going to lean towards cotrimoxazole to avoid any potential side effects from fluoroquinolones or tetracyclines, although some international guidelines are becoming a bit more open to short courses of doxycyclines for really serious infections.
All right, last section, let's talk prevention. If this bug is hanging out in our water systems, how do we stop it from infecting patients inside our hospitals? The battle is against a reservoir that's hiding in plain sight. So the main hideout is the hospital plumbing itself. Aeromonas caviae is a master at forming biofilms on the inside of pipes and drains, which acts like a shield, protecting it from standard chlorine disinfection. And the way it spreads is scarily simple. A splash from a contaminated drain onto a sterile field, using tap water to rinse a medical device, or even just direct contact. So what's the battle plan? Well, it's really a multi-pronged attack. It starts with a solid water management program, controlling temperatures and flushing outlets to prevent stagnation. It also means smarter engineering, like using deep sinks that don't splash everywhere. It means being absolutely militant about using sterile water for reprocessing things like endoscopes. And finally, it's about using the right precautions, standard for most cases, but you've got to step it up to contact precautions if you're dealing with uncontained diarrhea or a multi-drug-resistant strain. This all leaves us with a pretty big question to chew on. Aeromonas caviae is a perfect example of an environmental bug that found a happy home in our healthcare facilities. As that infrastructure gets older and older, what other microbes are just waiting in the wings to become the next major nosocomial threat? And just one more reminder, all the nitty-gritty details, the primary literature, and the full data set we used for this explainer are all in that source notebook document. The link is right there for you in the description. And that is our deep dive on Aeromonas caviae. If you found this useful, you know what to do. Make sure to subscribe for more explainers just like this one. And hey, we really want to hear from you. What complex topic in microbiology should we break down next? Drop your ideas in the comments below. Thanks for watching.